Right now, somewhere on Earth, someone is aging more slowly than you. Not because they deserve it, not because they optimized harder. Their cells simply handle stress better, and neither of you chose your version. Across Japanese, Italian, and American centenarian cohorts, researchers repeatedly found enrichment of specific variants of the gene FOXO3. These variants are associated with stronger cellular stress resistance, enhanced autophagy regulation, and more efficient DNA repair pathways. Published, replicated, statistically significant. In biology, that is the closest thing to applause. FOXO3 does not stop time. It regulates it at the cellular level. It tells cells when to repair, when to recycle damaged components, when to conserve energy, when to survive another wave of oxidative stress. Tiny regulatory differences. Decades of divergence in outcome. That is how longevity hides in plain sight. For all of human history, this advantage was random. Evolution distributed slightly different maintenance settings across humanity and called it fairness. Biology never promised equality. It promised variation. Now the rule is being challenged. A biotech company believes that this advantage does not have to remain a lottery. They believe it can be amplified through gene therapy. If evolution was blind, gene therapy is not. The company is Genflow Biosciences, a small-cap biotech, a FOXO3-focused gene therapy platform. An aged dog study already dosed, with no major adverse safety signals reported so far. A human program targeting metabolic liver disease, an aging-associated condition regulators can approve. No immortality slogans, just measurable biology moving quietly through development pipelines. And here is the line no one says out loud. The moment aging becomes adjustable, inequality stops being economic. It becomes biological. In this episode, we will uncover what FOXO3 truly does inside your cells, what Genflow is actually building versus what headlines exaggerate, why this is grounded molecular biology and not science fiction, where the real economic turning point hides, what realistically happens if this works, and who decides access if aging resilience becomes something you can enhance. Subscribe, because biology might be entering its software era, and software updates are rarely distributed evenly. Now let's open the genome and see what this gene is really doing inside you. To understand the ambition, we need to understand the mechanism. FOXO3 is not a miracle gene. It is a transcription factor. That means it does not build tissue. It does not create organs. It does not glow in the dark. It regulates other genes. It decides which cellular programs activate under stress. One of its primary roles is managing oxidative stress. Every time your cells produce energy, they generate reactive molecules capable of damaging DNA and proteins. FOXO3 activates antioxidant defenses and repair pathways before that damage becomes cumulative decline. It also regulates autophagy, the internal recycling system of the cell. Autophagy clears dysfunctional mitochondria and damaged components, preventing cellular clutter from turning into inflammation and metabolic dysfunction. When this process weakens, aging accelerates. When it remains efficient, resilience persists. FOXO3 interacts with insulin and IGF signaling pathways, systems deeply linked to growth, metabolism, and lifespan across species. It supports DNA repair, stabilizes cellular stress responses, and enhances survival under metabolic pressure, not immortality. regulation. This is precisely what longevity researcher Nir Barzilai, one of the leading scientists studying centenarians, has documented in long-lived populations. We explored his work in a previous episode. Long-lived individuals consistently show stronger metabolic protection, better cardiovascular resilience, and lower chronic inflammation. FOXO3 variants repeatedly appear within these protective profiles. Here is the shift. If longevity correlates with improved maintenance, then aging is not simply collapse. It is declining regulation. Evolution did not design immortality. It optimized maintenance. And maintenance is not fate. It is configuration. If aging is regulated, then in principle it can be tuned. Carefully, incrementally, technically. And that is the moment this story stops being observational biology and starts becoming engineering. Because maintenance can now be upgraded. In a sterile laboratory, a microscopic viral particle carries a genetic instruction that evolution never distributed equally. Not a new gene, not foreign DNA from a centenarian. An engineered vector designed to increase the expression of FOXO3, a regulator already present inside every human cell. This is gene therapy, specifically an adeno-associated viral vector platform, the same class of delivery system used in several approved genetic medicines. AAV vectors typically remain episomal, meaning they do not randomly integrate into the genome the way earlier retroviral systems once did. That reduces certain long-term genomic risks while enabling sustained expression in target tissues. The target is the liver, not by accident. The liver orchestrates lipid metabolism, glucose balance, inflammatory signaling, and systemic energy regulation. Age-related metabolic dysfunction often begins here. If you stabilize hepatic regulation, you influence the entire metabolic network. This is not cosmetic longevity. This is systemic leverage. Officially, the indication is metabolic liver disease linked to aging, not lifespan extension, not anti-aging. Regulators approve therapies for pathologies, not for existential upgrades. Framing matters. Biology is only half the battle. The other half is law. In parallel, Genflow Biosciences has completed dosing in an aged dog study using its FOXO3 construct. According to company disclosures, no major safety signals were observed during dosing. That matters. Gene therapy history has taught the field caution. Immune reactions, inflammatory responses, vector dose sensitivity, all of these risks must be cleared before efficacy even enters the conversation. And here is the critical line. There is currently no publicly available evidence demonstrating lifespan extension in treated dogs. No verified data proving longer survival. Efficacy results are pending. This is not a miracle announcement. It is a controlled experiment in enhancing resilience. It is not turning dogs into superheroes. It is testing whether regulation can be strengthened, whether cellular stress tolerance can be measurably improved, whether maintenance can be upgraded, and if boosting resilience works in disease, if metabolic stability improves, if inflammatory load decreases, the boundary between therapy and enhancement becomes fragile. Because the moment regulation becomes adjustable, aging stops looking like inevitability and starts looking like configuration. And once configuration is possible, the question is no longer whether we can intervene. It becomes how far we are willing to go. Natural genes are not patentable. Courts have made that clear. You cannot claim ownership over a DNA sequence that evolution already wrote. FOXO3 belongs to biology, but methods are patentable. Viral vectors are patentable. Manufacturing platforms, purification systems, tissue targeting strategies, and specific disease indications can all be protected. You cannot patent nature, you can patent the upgrade. If a validated method to enhance FOXO3 expression proves safe and effective, the intellectual property will not sit in the gene. It will sit in the delivery platform. That is how modern biotechnology works. Value concentrates in execution. Now imagine the first approval, a defined medical indication, metabolic liver disease linked to aging biology, clear clinical endpoints, reimbursement pathways, controlled rollout. The therapy enters the system not as immortality, but as medicine. Early gene therapies today can cost hundreds of thousands, sometimes over $1 million per patient. Manufacturing viral vectors is complex. Capacity is limited. In the beginning, access follows capital. That is not dystopian. It is how nearly every advanced medical innovation has started. But history shows a pattern. Sequencing the human genome once cost billions. Today it costs less than a high-end laptop. mRNA platforms went from experimental to global deployment in record time. The first computers filled rooms. Now they sit in your pocket. Biotechnology scales when demand, capital, and manufacturing converge. If cellular resilience can be enhanced safely, the first wave will be clinical. The second wave will be optimization. Over time, production scales, costs fall, and access widens. That is not guaranteed, but it is the historical trajectory of transformative technologies. The first generation that can afford enhanced stress resistance may biologically diverge from the rest. That is a real possibility, but divergence is not destiny. Markets expand, infrastructure matures. What begins exclusive often becomes foundational. Vaccines were once rare, insulin was once scarce. Both reshape civilization. So the real turning point is not whether resilience can be patented. It is whether society chooses to scale it responsibly. For the first time, aging could become a distribution problem, and distribution problems can be solved. Let's slow down. No immortality headlines. No 300-year lifespans. A realistic scenario, if this works, begins quietly. Improve liver function in patients with metabolic disease. Better regulation of lipid metabolism. Lower inflammatory markers. Measurable stabilization rather than biological fireworks. If enhancing FOXO3 expression strengthens stress response pathways, we would expect improved cellular housekeeping, more efficient autophagy, slightly better DNA repair efficiency, reduced oxidative burden over time, not reversal of aging, improved management of it. Health span could extend modestly. 5 years of improved metabolic stability, 10 years of reduced inflammatory drift, fewer complications cascading from age-associated dysfunction. That is not science fiction. That is incremental physiology. And we must stay precise. Aging is multifactorial. Telomere dynamics, epigenetic drift, mitochondrial dysfunction, stem cell exhaustion, proteostasis collapse, immune remodeling. FOXO3 is one regulatory node inside a vast network. One lever, not the control panel. But here is where the future bends. If even one component of aging can be tuned safely, biology shifts category. Medicine moves from repairing failure to optimizing resilience. The objective changes. Not survival after breakdown. Sustained stability before breakdown. The revolution may not look dramatic. It may look like better blood tests, slightly improved biomarkers, slower metabolic deterioration curves, less chronic inflammation at 50, less cardiovascular fragility at 70, quiet curves bending in spreadsheets before they bend in cemeteries. Small shifts compound across populations, later retirement ages, longer productive decades, health systems under less strain, extended periods of cognitive clarity. When average decline slows even slightly, entire economic models adjust. And once society sees that biological parameters can be adjusted, even modestly, the psychological shift becomes irreversible. Aging stops being a fixed narrative and starts being a variable under study. That does not mean unlimited control. It means directional influence. Because history shows something consistent. When a system becomes measurable and modifiable, it becomes optimized. And optimization, once it begins, rarely stops at the first success. Even small biological shifts reshape civilization. We started with a simple observation. Some people are born with slightly better cellular resilience, random advantage, molecular luck. Now we are approaching a world where resilience may no longer be random. If aging becomes even partially engineerable, the question stops being biological and becomes philosophical. Is it a therapy? Is it a luxury? Is it a right? Is it an upgrade? For the first time in history, we are not just treating disease. We are negotiating with biological decline, not erasing it, not conquering death, but adjusting the slope of deterioration. That shift is not apocalyptic. It is monumental. Every major medical advance once felt unnatural. Vaccination altered immune destiny. Antibiotics reshaped survival curves. Organ transplantation redefined viability. At first controversial, then regulated, then normalized. Progress rarely announces itself as comfortable. It becomes comfortable later. If safe gene-enhanced resilience becomes available, distribution will define the era. Should it be integrated into universal healthcare systems as preventive infrastructure? Or should it begin as market-driven access that gradually scales with competition and manufacturing maturity? This is not about immortality. It is about whether reducing long-term biological fragility becomes a shared public good or a staged technological rollout. History suggests that what begins expensive often becomes standard. The question is not whether scaling happens. It is how deliberately we design it. Because once decline becomes adjustable, society changes its relationship with time. Career planning shifts, education timelines shift, retirement shifts, health systems shift. When biology becomes tunable, culture follows. So here is the question no one can avoid. If a safe, clinically validated gene-enhanced resilience therapy became available tomorrow, would you take it? And should it belong to everyone or to those who can afford to move first? Tell us what you think in the comments, not in slogans, in reasoning. Because the future of longevity will not be decided by scientists alone. It will be shaped by voters, regulators, investors, and people like you. Progress is never just invented. It is negotiated.